Hi friends, welcome again. Hope you could solve the problems in my last video on DNA structure. Today we will be learning principle and technique for Sanger's DNA sequencing through problem-based learning. So there will be a few problems. After understanding the basics, you will try those problems and will check and match with my answers. Since we understood the DNA structure and also attempted some problems, let's first look at the answers and explanations for problem 1. The answers are simple, they are just fill in the blanks. For problem 2, it's easy to calculate the length of 128 base pairs if we know the length of one base pair, which is 0.34 nanometer. Hence the answer will be 43.52 nanometer for 128 base pairs. For problem 3, we need to keep in mind that number of cytosines will be equal to number of guanine and number of adenine will be equal to number of thymine bases. So let's first calculate how many cytosine bases are present in each strand and then add up for a total which is 183. It means 183 cytosine bases and 183 guanine bases are present in this fragment. Since fragment length is 300 base pair, we subtract 183 bases to get the number of adenine and thymine bases, which is 117 bases each. Now since cytosine and guanine pair with 3 hydrogen bonds and adenine and thymine pair with 2 hydrogen bonds, we need to multiply 183 with 3 and 117 with 2 and then add them up. The total number of hydrogen bonds will be therefore 783 in this 300 base pair fragment. For problem 4 and 5, which are MCQs, the answers should be simple. Hope you learn to solve such problems. In next step, we look at the problem based on Sanger sequencing technique. Have a look at this problem where 4 reactions have been given and you have to determine the DNA sequence from the given data. Is it confusing? Never mind. Let's learn the Sanger sequencing principle and technique first. Frederick Sanger, a British biochemist, won 2 Nobel Prizes for giving us the protein sequencing and DNA sequencing technique. His DNA sequencing technique is still a gold standard, though it has been automated now and several next generation sequencing techniques are also available in the market. He created special types of DNA nucleotides known as DI-DEOXY nucleotides. DI-DEOXY means that at 3' position of the deoxyribose sugar, also there is a missing oxygen, which disturbs the OH group, which is very crucial for creating a phosphodiester bond by the DNA polymerase. Now since DNA polymerase cannot create the phosphodiester bond with the next nucleotide, the chain will terminate and we will get unique sized PCR products. Therefore, this method is also known as DI-DEOXY or chain termination method. Here is an unknown DNA template whose sequence we need to find out. For demonstration's sake, I will use the sequence. Sequencing process is very similar to PCR reaction and DNA replication process. Here you see a primer which is required to provide a 3' OH group to DNA polymerase, which then extends this primer to make new DNA. Primer is just a small piece of synthetic DNA around 18 to 30 base pair long. The primer sequence has to be complementary to the template DNA. Now question arises that if we do not know the sequence of template DNA, how can we design a primer? The solution to this problem is that our template is first cloned in a sequencing vector, which is designed and genetically engineered. So we know its sequence and hence can design a primer against one strand of this vector, which can be extended to cover the template sequence thereafter. In next generation sequencing, instead of sequence vector, adapter sequences are used whose sequence is also known to us. Then we have 4 DNTPs or nucleotides, which are the building blocks for the new DNA strand or PCR product, and 4 types of DDNTPs as well. We begin the sequence reaction in 4 separate tubes. Add a certain amount of single standard template DNA containing multiple DNA fragments, primer, all the 4 DNTPs including DATP, DCTP, DGTP and DTTP, which are normal nucleotides. Also add DNA polymerase enzyme and buffer, etc. Most importantly, each tube receives a certain concentration of only one type of DDNTP as indicated here. Now the reaction starts and DNA polymerase starts extending the 3' OH end of the primer to create complementary DNA sequence. The direction of new DNA sequence will be always from 5' to 3'. Let's take the given template and write it as 3' to 5'. So we can see the primer extension reaction from 5' to 3'. In the first tube, for all the thymines or T bases present in the template sequence, we need A base. This tube has 2 types of A nucleotides, normal DATP and DDATP. Based on probability, the new sequence can incorporate a DDATP or DATP against any of the T base in template, as DNA polymerase cannot make out the difference while picking up the nucleotide. However, it can't extend the polynucleotide chain further. If a DDATP has been added, it can't extend. Hence, the new DNA sequence may terminate at various locations of T, as shown here, and we get a unique set of PCR product sizes, like here 1 base pair 9, 14, 20, 21, 23 and 27 base pair products have been shown. Similarly, different PCR products we get from DDCTP tube and DDGTP tube and from DDTTP tube. Now we run all the PCR products from the 4 tubes on a large urea gel, which is made up of polyacrylamide and can resolve PCR products differing in just 1 base as shown here. Let's learn how to read the PCR product sequence from here. We will begin from the smallest size product at the bottom and move towards the bigger size products on the top, counting across all the 4 lanes combined as shown here by arrows. Now our PCR product sequence will be 5' to 3' as given here. Since we are reading the PCR product sequence, our template DNA sequence will be complementary to it. This will be from 3' to 5' and then we have to revert it back to 5' to 3'. So this is our original sequence. You can also see a real picture of the Sanger sequencing gel. In this process, for band detection, radioactive DDNTPs and autoradiography was used by Sanger, which has now been replaced by fluorescently labeled DNTPs. Original Sanger sequencing was a laborious process. So in 1987, Applied Biosystem Incorporation converted this technique into a fully automated process. Now 4 differently colored fluorescently labeled DDNTPs are used, which can be identified easily on a chromatogram. Instead of a large urea gel, we use capillaries filled with polyacrylamide. We can use 96 capillaries simultaneously. So we can process 96 samples in one run. When PCR products move through these capillaries, lasers fall on them and activate the fluorescent labels, which are then detected and converted into a chromatogram. So friends, we learned some basics about Sanger sequencing. Now you can solve these problems and can either provide answers in comment section or check for answers given in my next video. Hopefully you liked today's tutorial and learned something new. If you like the problem-based learning, based approach, please like and subscribe to my channel so that I can keep making new videos for you. My next video will be about restriction enzymes and restriction mapping. Don't forget to watch. Till then, thank you and bye bye.